So are you watching my screen? Okay. Yes. Thank you. So hi everyone. I'm Kenichiro Motomura from maternal fetal immunobiology unit of the perinatology research branch. I'm very happy to be here, and it's my pleasure to be presenting our work. So this is the process of reproduction. As an obstetrician, we take care of pregnant women and their delivery. However, this is a sequential process, and it is very important to understand that any problems during pregnancy can affect the life of their babies. Indeed, pregnant women can experience multiple types of pregnancy complications or pregnancy-specific disorders. In particular, great obstetrical syndromes listed here share unique features, including multiple etiologies, long preclinical stage, frequent fetal involvement, clinical manifestations, which are often adaptive in nature, and gene-environment interactions that may predispose to the syndrome. And also today's topic is pregnancy and inflammation can be included in most of these great obstetrical syndromes. And among these, preterm birth, defined as delivery before 37 weeks of gestation, is the leading cause of neonatal morbidity and mortality worldwide. Preterm birth is often preceded by spontaneous preterm labor, a syndrome of multiple etiologies. Among these, intra-amniotic infection inflammation is causally linked to preterm labor. Indeed, if we classify the types of preterm labor by inflammatory status of amniotic fluid, 40% of them are either microbial associated, also known as intra-amniotic infection, or sterile intra-amniotic inflammation. So what is intra-amniotic infection? Here you can see the pregnant uterus. Amniotic fluid surround the fetus. And you can see placenta and fetus here. And this part is called cervix. So, So intra-amniotic infection is caused by the microbes ascending from the lower genital tract. They causes the infiltration of fetal leukocytes to the amniotic cavity and facilitate the release of cytokines such as IL-6, IL-1 beta, and TNF. Among these, interleukin-1 beta is referred to as a master regulator of onset of labor, as demonstrated by several groups. So interleukin-1 beta is a zymogen that requires enzymatic cleavage to display its bioactivity. A primary mechanism underlying this process is the NLRP3 inflammasome. When pathogen-associated molecular parents, or PAMPs, is sensed by pattern recognition receptors such as toll-like receptors, it facilitates the translation and transcription and translation of NLRP3 inflammasome components such as NLRP3, ASC, or pro-caspase-1 and pro-interleukin-1 beta. The assembly of NLRP3 inflammasome activate the caspase-1 by self-cleavage, and this active caspase-1 cleave pro-IL-1 beta to mature IL-1 beta, which is released to the extracellular space and induce strong inflammation. So we have generated considerable evidence suggesting the role for the NLRP3 inflammasome in physiologic labor as well as preterm labor. However, the molecular underpinnings of preterm parturition resulting from intra-amniotic inflammation, as well as the fetal and maternal contribution are poorly understood. So therefore, the first question we investigated in this study was whether NLRP3 inflammasome is essential for the premature onset of labor induced by intra-amniotic inflammation. Before moving on to the results, I'd like to emphasize that intra-amniotic inflammation is a local inflammatory response and subclinical in nature. Therefore, coincidence of maternal systemic inflammation is rare. To mimic this local inflammatory response, we have generated a translational model of intra-amniotic inflammation using mice. You can see a pregnant mouse. High power shows one amniotic sac. We injected gram-negative bacteria product lipopolysaccharide to the amniotic sac under ultrasound guidance. So you can see here the fetus. This black area is the amniotic cavity, which filled with amniotic fluid. The needle is here, and then you can see the jet sign of indicating the successful injection of LPS to the amniotic fluid. So we utilized this technique to NLRP3 deficient, as well as wild-type mice in late pregnancy, and perinatal outcomes such as rate of preterm birth and neonatal mortality were monitored. So consistent with our previous study, most of the wild-type mice injected with LPS delivered preterm, which was reflected by their reduced gestational lengths. And strikingly, only a small proportion of NLRP3 deficient mice delivered preterm, indicating that NLRP3 signaling is required for the premature onset of labor. Similar to pregnancy outcomes, neonates born to wild-type dams injected with LPS died shortly after birth, whereas those born to NLRP3 deficient dams displayed a mortality rate similar to controls. Therefore, NLRP3 deficiency protects against intra-amniotic inflammation in this preterm birth and neonatal mortality. So given that preterm birth is causally linked to intra-amniotic inflammation, we next wondered whether NLRP3 inflammasome activation is involved in inflammation of the amniotic cavity. We intra-amniotically injected NLRP3 knockout and wild-type mice with LPS and collected amniotic fluid to determine inflammasome activation. So we observed caspase-1 activation and mature interleukin-1, increased level of mature interleukin-1 beta in wild-type mice, but was not observed in knockout mice. Therefore, NLRP3 deficiency impairs inflammasome activation in the amniotic cavity. So we next investigated whether NLRP3 is implicated in fetal inflammation triggered by intra-amniotic LPS. So we know that a clinical sign of fetal inflammation is alteration of the fetal heart rate. Therefore, we evaluated the fetal heart rate in knockout and wild-type dams injected with LPS using ultrasound. We observed that fetuses of LPS-injected dams were bradycardic regardless of NLRP3 expression. However, knockout mice are even lower heart rate. This suggests that fetal well-being was compromised in response to LPS, even if preterm birth did not occur. Moreover, we evaluated the fetal inflammatory responses, given that intra-amniotic inflammation damages the fetus through neonatal sepsis, brain, lung, intestinal injury. Based on this clinical observation, we evaluated inflammation in the fetal lung and intestine in this study. So we observed that LPS injection induced an inflammatory response in the fetal lung in both wild type and knockout mice. The fetal intestinal inflammation was less evident than the fetal lung. Thus, NLRP3 deficiency impairs inflammasome activation in the amniotic cavity, but not fetal inflammation. This suggests that fetal inflammation alone is insufficient to initiate preterm labor and birth. And indeed, labor requires the coordinated activation of multiple organs, including fetal membrane activation, as well as uterine contractility and cervical dilation. They are called the common pathway of labor. Therefore, we next explore whether NLRP3 deficiency interferes with the common pathway of labor. We intra-amniotically injected LPS to NLRP3 knockout in wild-type mice and collected tissues participating in the common pathway of labor. So first, we found that LPS induced caspase-1 activation in the fetal membranes, which was abrogated in knockout mice. Consistently, mature interleukin-1 beta was increased in the fetal membranes of wild-type mice, but not in those of knockout mice. As you can see here, the immunoblotting and immunofluorescence staining. Thus, NLRP3 deficiency protects against preterm birth by interfering with the process of fetal membrane activation, which includes inflammasome activation. So in the uterus, LPS induced the upregulation of contractility genes in wild-type mice. However, such an increase did not occur in knockout mice injected with LPS. LPS also induced caspase-1 activation in the uterus of wild-type mice, which was abrogated in knockout mice. Moreover, the production of mature interleukin-1 beta, which was increased in uterus of wild-type mice, was not observed in knockout mice. These data show that NLRP3 deficiency also protects against preterm birth by interfering with the process of uterine contractility, which includes inflammasome activation. Next, we evaluated the third component of the common pathway of labor, cervical dilation. We measure cervical width as a surrogate for cervical dilation and found that LPS induced cervical dilation in wild-type mice. However, such dilation was reduced in knockout mouse, so we measured the part of this in cervix. And consistent with our uterine findings, LPS induced the upregulation of contractility gene expression in the cervix of wild-type mice, but this was not observed in knockout mice. However, inflammasome activation was not observed in the cervix of wild-type or knockout mice in response to LPS, which is consistent with a previous study. These results suggest that NLRP3 deficiency protects against preterm birth by interfering with the process of cervical dilation in the absence of inflammasome activation. Together, these data indicate that NLRP3 deficiency protects against preterm birth by interfering with the common pathway of labor. And another important component of the labor process is the infiltration of leukocytes into the uterine and decidual tissues. Therefore, we next investigated whether NLRP3 regulates leukocyte infiltration in these tissues. We isolated leukocytes from the uterus and decidua of wild-type and NLRP3 deficient mice injected with LPS and performed immunophenotyping. So LPS injection increased the number of neutrophils in both wild-type and knockout mice. The number of macrophages were unchanged by LPS injection in both genotypes. Therefore, NLRP3 signaling is not implicated in neutrophil infiltration in these tissues. So we next explored whether the functions of these innate immune cells were altered using cell sorting and RNA sequencing. So in the decidual, neutrophils from knockout mice displayed gene expression that was enriched for host response processes such as defense response to bacteria. On the other hand, uterine neutrophils displayed gene expression enriched for cytokine signaling processes such as regulation of IL-1 production. And macrophages in the decidual of knockout mice displayed altered gene expression with enriched of processes such as positive regulation of leukocyte differentiation. In the uterus, such cells showed gene expression associated with cell migration. So together, these data indicate that NLRP3 regulates neutrophil and macrophage functions in a tissue-specific manner. So up to this point, we have shown that participation of NLRP3 signaling in maternal and fetal compartments. So actually, the fetal membrane activation is belong to the fetus, and uterine contractility and cervical dilation was done by maternal tissues. Therefore, we last evaluated the maternal and fetal contributions to NLRP3 signaling. So to address this question, we utilized embryo transfer. So we collected the embryos from wild type and knockout mice and transferred them to the pseudopregnant animals of opposite genotypes. And then we obtained dams, wild-type dams carrying knockout fetuses and knockout dams carrying wild-type fetuses. And we injected LPS intra-amniotically for these mice. And as expected, the majority of wild-type mice carrying wild-type fetuses delivered preterm upon LPS injection. And notably, knockout dams carrying knockout and knockout fetuses deliver preterm, but small number of them are delivered preterm. And importantly, knockout dams carrying wild-type fetuses and wild-type dams carrying knockout fetuses both showed reduced rate of preterm birth. You can see here and here. And indicating that both fetal and maternal NLRP3 signaling contributes to preterm parturition induced by intra-amniotic inflammation. So collectively, our data show that NLRP3 inflammasome activation triggers intra-amniotic inflammation, leading to preterm birth and neonatal mortality. NLRP3 deficiency impairs the common pathway of labor and dampens the intra-amniotic inflammatory response that is responsible for the mature onset of labor and neonatal mortality. Moreover, these mechanisms are mediated by both fetus and the maternal tissues. So I'd like to thank the member of the Gomez-Lopez lab, especially my mentor, Dr. Nari Gomez-Lopez. I also like to thank our collaborators and acknowledge our funding sources. And so I stop here and then happy to have any questions. Thank you very much.
Thank you very much, Kenichiro. And I was wondering if anyone has any question. If not, if people join, before people start asking any questions, I guess I will start with mine. So I was wondering if you see the same phenotype in an heterozygous mice of NLRP3, having just one copy. You see the same phenotype, or you have to deplete both of alleles to have a phenotype there?
Oh, I see. You are asking the mating of wild type and knockout.
Yeah.
So we also showed this mating pair result in the paper, and then actually the both if the fetal genotype is a heterozygous, the rate of preterm birth was reduced.
Okay.
Yeah.
My second question will be mostly related to if there's any approach to give inhibitors of the inflammasome in pregnant women who have risk, or if there are any ongoing trials to try to move this data to the clinics.
Actually, that's a very important question, particularly for physicians. And we have already showed that the NLRP3 inflammasome inhibitor, which is called MCC950, could be inhibit the preterm birth in mice. However, currently we don't have the information for the safety of this reagent to the pregnant women. So we don't have any ongoing trial for that, but I think it's a promising strategy for preventing preterm birth in the future.
And I guess finally, just to close up of this topic, like, is there any GWAS analysis of any sort of type of mutation that have an enhanced inflammasome activation and that could be a risk for having failed pregnancies in women?
Yeah, that's also an interesting question because we can see the phenotypic change in humans by your mentioned people, by analyzing the people you mentioned. And but unfortunately, I don't think we have the information of pregnancy of these patients. Actually, there's a patient with the mutation of NLRP3, and they have severe inflammatory responses. But I don't think we have the information of pregnancy because I think they have a severe inflammation and maybe they couldn't get pregnant. But it's an interesting topic.
Well, yeah, thank you very much. I don't know if there's any more questions in the chat. Either way, I think.
Yeah.
Hey, Eugenio?
Yes, Nuri?
Yeah, I have a question.
Yeah, okay.
Hey, Kenichiro, it's great work. I really like the presentation was amazing. So I have a question about your immune cell compartment. So you showed the neutrophils and the macrophages. Did you find other cell types? I don't know, thinking about maybe NK cells that are involved in this part. I know they're involved earlier in pregnancy, but I know about in late pregnancy, the role of NK cells in this part, or maybe other leukocytes.
Thank you for your question. So actually, we did immunophenotyping. So we included T cells, B cells in our panel, but we haven't included NK cells because it's already reported that NK cell was not increased in this type of inflammation. And then also T cells and B cells, we didn't observe the increase of infiltration to the decidua and uterus.
Okay, and my second question is about the macrophages. So are these M2 or after the LPS challenge, should they turn to another like an M1 type of phenotype?
Yeah, in this study we haven't distinguished M1 and